Hello, everyone. Welcome back. Today we're talking about Chapter 16, and we're going to talk a little more about genes and how they work. Now, we've talked about genes before. We've mentioned it in our Mendelian genetics, but we're going to kind of connect genes to this central dogma, which we've talked about briefly in the very beginning of this course. But we're going to talk about genes, their role in this central dogma, and how do mutations affect this flow of information. At this point, you are no stranger to DNA, the blueprint for life, right? Your instruction manual, your genes. And we now know more about DNA, about how that DNA is passed from generation to generation, how chromosomes work and all this stuff. But we still didn't really understand gene expression. Okay, how do genes work at a molecular level? And how do we get that information from DNA to a fully functioning product, right? How do we get the instructions for blue eyes from a sequence of bases to an individual with blue eyes? What is that process? What is that pathway? And we decided we could figure out what genes do by breaking them. Okay, we could take a gene, we could damage it to the point where it's no longer working, and then observe what happens to the phenotype. Okay, how are they connected? And anytime we break a gene, we call a non-functioning allele a null or loss of function allele. Now, what they did to damage genes, they worked with some bread mold, and what they did to it is they just blasted it with radiation. Okay, irradiated it up to the point where certain genes were no longer functioning. And once they did that, they looked for mutants, right? Things that could not make certain compounds. And they were successful. They found that they did, through this process of blasting bread mold with radiation, they created mutants with defects in certain genes. And all of this led to a one gene, one enzyme hypothesis. Okay, each gene contains the information to make a certain enzyme, one single enzyme. However, we later figured out it wasn't just enzymes. Okay, a lot of your DNA, yes, does make proteins, does make enzymes, but that's not the only thing that your DNA contains the recipe for, okay? It can make more than just proteins and enzymes. So we now call that one gene, one polypeptide. So we just made it a little more vague. So genes specify the production of proteins. But how can they do this? How do we get from one to another? And it all has to do with the structure. Okay, Watson and Crick's model, well, really Rosalind Franklin's, but whatever. Watson and Crick's model didn't really explain how DNA could directly make proteins. Okay, how does it work? That is not, DNA's structure would not allow it to also act as a catalyst. Okay, it has very regular structure. So how do we make such a wide variety of different proteins from DNA? And that is where we came up with the genetic code hypothesis. Okay, the sequence of bases in DNA act as a code. Okay, so DNA is not what's physically making proteins. DNA is just the information storage. Okay, what combination of those bases specify what combination of amino acids we're going to see. So just like letters in a word, right, the order of our bases is going to code for a specific amino acid. Okay, that's what we're saying here. And a particular stretch of DNA, which remember we call a gene, is going to specify the sequence of amino acids in a protein. Okay, but the information from DNA is not going to be directly used to make a protein. Okay, there is going to be some sort of in-between. Okay, and part of the reason why is location. Okay, we have kind of a location issue, right? If we're talking eukaryotes, where is our DNA stored? In the nucleus of our cell. And what is our protein factory? Ribosomes. And most of our ribosomes are going to be found either in the cytoplasm of our cell, or they are going to be found in organelles like the rough ER. So we have a location issue, right? The recipe is locked away somewhere other than the kitchen, right? We need to figure out a way to get that recipe where it needs to go. And that is where we found messenger RNA. Okay, this is the in-between. This is our information carrier. It's going to make a copy of the recipe of the DNA, and it's going to bring it to the ribosomes. It's going to bring it to the areas where protein synthesis occurs. Okay, and with this, we're going to see a very specific enzyme called RNA polymerase. This is what's going to make that mRNA. Very similar to DNA synthesis, little different. It's going to use our DNA strand as a template, and it's going to make a single mRNA strand that is going to serve as our message. Okay, that's what can leave the nucleus and go and actually make a protein. So that flow of information, DNA to mRNA to protein, that is our central dogma. Okay, this flow of information. And mRNA was that connector. It was the missing link between DNA and protein. So that's our central dogma, our kind of unifying theory of molecular biology. DNA to RNA to protein. Right here. Okay, this is our central dogma. Now we're going to add some things to this central dogma. So this isn't our finished central dogma, but this is the most simple version, DNA to RNA to protein. Okay, genes are stretches of DNA that code for proteins, but they do it through mRNA. So DNA sequence makes an mRNA sequence, and then our messenger takes that message and codes for a sequence of amino acids that make a specific protein. So those processes, transcription and translation. I always like to ask my students, what does it mean to transcribe something? Right? You might have heard of it in like court TV shows, right? Court reporters transcribing something. But essentially, if you're transcribing, you're writing down a copy, right? You're taking down a copy. And that's exactly what transcription is doing. We're using DNA, our information storage, and we're making a copy. We're making our messenger RNA. So that process from DNA to RNA is transcription. Now, what does it mean to translate something? If you are bilingual, you very likely translated something at some point in your life, right? You've taken it from one language to another. You haven't changed what you're saying, right? The meaning still stays the same, but you're taking it and you're moving it from one language to another. And that's what we're doing during translation. We're taking this sequence of bases, these nitrogenous bases, and we're making it into amino acids. Okay, we're taking our message and we're translating it into a protein, into a series of amino acids. So this is our new updated central dogma. We're not quite done yet. We're still going to add some more. Okay, but now we've identified what these arrows mean, right? DNA to RNA, transcription, make a copy. RNA to protein, translation. Now, how does genotype and phenotype fit in? Remember, genotype is what alleles do you have, right? Are you homozygous dominant? Are you heterozygous? Or are you homozygous recessive? Okay, what alleles do you have? What do your genes look like? But remember, this is all determined by the sequence of bases in your DNA, right? What alleles you have are all determined on this code, this genetic code. What bases do you have and in what order, right? So that's our genotype, sequence of bases. And then phenotype is the organism's physical appearance, right? Blue eyes versus brown, whatever. But this phenotype is usually a direct product of what proteins are produced. So I like to modify this flowchart because I don't think this should be an arrow here. I think it should just be a little parentheses, so we're going to change that. Now we've updated our central dogma, right? DNA, or the genotype of an individual, okay, through transcription, okay, becomes an mRNA messenger, and that mRNA messenger through translation still going to abbreviate transcription and translation, becomes a protein, and that protein is directly responsible for an organism's phenotype. So, for example, blue eyes is the absence of the protein melanin in one's eyes. So, because of your genotype, because of a certain mutation, okay, we get melanin not produced. So we've changed our protein product, and blue eyes is the result. Okay, we've altered our phenotype because of changes to our genotype. Okay, so this is our full central dogma, okay, including genotype, including transcription and translation, and phenotype. So this is what I recommend that you know for our upcoming exam. Now, here is just showing that flow and showing changes, right? So we have our original sequence, transcription, we get our message, translation, we get our protein, our series of amino acids, and you'll see that this sequence is responsible for coat color in mice. Okay, so maybe melanin is produced and we have a nice dark coat in our mouse. Now, say we have a mutation, some sort of change in our bases, in our genotype, changes our message, changes our protein, okay, and our phenotype is affected. Now we don't have as much of that pigment, and we have a much lighter coat color for our mouse. Now, are there any times where this central dogma is changed or altered? Okay, there are two, in fact, two specific times where our central dogma is not what we just previously described. Okay, our first is that sometimes with our flow of information, okay, it stops at RNA. Okay, we have a lot of different RNAs in the body, not just messenger RNA, okay, and they do important functions, they do different jobs. Okay, so sometimes our flow of information can stop right from DNA to RNA, and then those RNA molecules are going to go and do their job, do some other function in the cell. Okay, they will never be translated into proteins. So that's one example of where our flow is a little different. Now, the second example is a little more unique. Okay, this is where we actually reverse the flow of information. We can take RNA, and from RNA, we can make DNA. And this is through a very specific enzyme called reverse transcriptase. Essentially, we're reversing transcription. And this enzyme is found in some viruses that need to use an RNA template to make more DNA. So you'll notice our flow of information in this case is reversed. Now, when we talk about the genetic code, this is going to specify how do we read our recipe, our series of nucleotides, and how does that relate to amino acids. And for each word in our genetic code, okay, this is going to be a series of 3 bases. And we are always going to see it in a series of 3. Okay, we call this a triplet code. So that just means when we're reading our DNA strand, we're always going to read it in nice little series of 3. Okay, and that series of 3 we call a codon. And the reason why we read it in a series of 3 is because each codon is going to code for a specific amino acid. Okay, so this series of 3 we call the reading frame. So I'll draw it again here. some series of 3 like that. Okay, now you'll notice what happens when we say delete a single nucleotide from our reading frame. Look what happens. Okay, now our reading frame is destroyed, right? Now we have an entirely new codon, and we have a codon that's missing one at the end. So we've changed the way we read this entire code if we add in 1 or 2 bases. However, if we add in just any series of 3, okay, if we add in another 3, A, C, C, okay, our reading frame is not destroyed. Our first 2 remain just fine. No alterations to our codon. We just have an extra codon that gets added on or added in. So this is how we decided that, yes, these in triplets, okay, because anytime, once again, we add anything that's not a triplet, we really mess up how we read this sequence. It was Nirenberg and Leder who were the ones who determined what codon codes for what amino acid. Okay, and we're going to see they made a lovely large chart that you are going to become very familiar with in both lecture and lab. But they were not only able to identify what codes for what amino acid, but they identified a few very important types of codons. So one is a start codon, and this one is always AUG. Okay, and we're talking mRNA. And this AUG is always going to code for a methionine. And this is what's going to tell translation to start. Okay, this is where we need to start reading our message, okay, the start codon. Now, once we are done, once that message is over and we need to stop making our protein, okay, that's when we run into a stop codon. And there are 3 different stop codons, but just like the name suggests, this is going to say, we're done. Stop reading the message, stop protein synthesis. Now, the other 60 codons that we're going to run into are going to code for some sort of amino acid. And remember, there's 20 different types of amino acids. Okay, so here is that lovely chart I was telling you about earlier, and we're going to kind of walk through how we would do a transcription and translation problem. Okay, so you'll notice I have given you a template DNA strand. Okay, I've indicated direction because that will be important. And you'll see I have my bases outlined in series of 3, so our codons. Now, can I just use this chart directly? No, right? Because we do not translate from DNA, right? We have to make a copy first, right? mRNA. We have to transcribe our sequence. So this is where our base pairing rules come into play. Okay, so I'm going to write it out just like this. A pairs with what? T. So our first 3 are 3 T's. Okay, G pairs with what? C. Okay, and C pairs with what? G. So G and G. And T pairs with what? A. A pairs with what? Excuse me, T pairs with what? A. And A pairs with what? T. Okay, are we good? No, we are not good, because are there any T's in mRNA? No, we don't have any thymine in RNA. Remember, thymine is replaced with uracil. So once I have made my transcription with my base pairing rules, I'm going to go in and I'm going to replace all the T's that I had with U's. Okay, because uracil replaces thymine. Okay, so there we go. This is my sequence of mRNA. Now I can finally use this chart to figure out my protein, okay, amino acid. Okay, what does my protein look like? And remember, I'm going to read in series of 3. So, okay, U. First, I would look over at my first base and I'd say, okay, it's a U. That's what my first base looks like. So I know I'm going to be in this row right here. Now my second base over at the top, okay, my second base also a U. So now I know I'm going to be in this column right here. And then if we look within, you could look over to the third base, but at this point I usually just look into that little section of our chart, and you'll see UUU codes for phenylalanine. So that is our first amino acid, and I'm keeping with my directionality, right, because that's important. Phenylalanine. Boom. Okay, our second codon, CGG, and we'll see our first base, C. Okay, second base is G, and we got CGG right here. Okay, so that is arginine, ARG. Okay, and then our third base, AAU. Okay, first base A, second base A, and AAU right here, asparagine, ASN, and 3'. Okay, so this is our protein sequence. This is our series of 3 amino acids that makes up our protein that is created from this template DNA up here. So make sure you know how to do this. Make sure if I give you template DNA, you are able to transcript and transcribe that into a final protein product using this chart. Now, some things we need to know about the genetic code. Okay, 1, it is redundant, which means all but 2 amino acids are encoded by more than 1 codon. So let's look at our code. You'll notice there's 4 different codons, and they all code for leucine. Okay, that's what we mean by redundant. Okay, multiple different codons code for the same amino acid. This code is also unambiguous. You're never going to see one code for more than 1. So what that means is if we have, say, CCU, okay, CCU is always going to be proline. You're never going to see that series of 3 code for anything else. It's always going to be proline. It is also non-overlapping. We always read it 3 at a time. Okay, 3 bases at a time and 1 codon at a time. Okay, so we're never going to see it overlap. It's always going to be in that nice little series of 3, just like we always write out. Okay, now another thing, our code is nearly universal. And this one's kind of cool. It's basically the same in all organisms, okay, whether it be you or a snake or a bread mold or whatever, okay, this code still applies. This chart still applies. Okay, and this is one of our greatest evidences for a common ancestor between all organisms, right? Why do we have this universal genetic code unless at some point we all came from the same ancestor? So, very few minor exceptions to this, nearly universal. Lastly, our code is also conservative. Okay, you'll notice if we look at our codons, right, let's do proline again. The first 2 are always identical. CC, okay, all CC is going to be proline, and you'll notice it's just that 4th base that changes. Okay, that's what we mean by conservative. Most of the time, you're going to see these first 2 bases be identical for that codon. So serine's always UC, just that last base that changes. Let's see, another valine over here is always GU. And you'll see there's some areas where this is not always true, like with GA or with AG. But for the most part, a lot of our bases are conservative. The first 2 are usually identical. Now, as we've already demonstrated, we can use this code and we can use the central dogma to predict. We can take a region of DNA and we can figure out what amino acid sequence is going to be made. But if we wanted to flip that around, we could also figure out what mRNA and DNA sequence that would code for an amino acid. So imagine if you had a protein, you could kind of reverse engineer that protein and generally figure out what the DNA sequence looks like that made it. However, because of redundancy, you're gonna get a little bit of ambiguity because more than one sequence could code for the same amino acid sequence. So here is a little bit of practice. This is showing you exactly what we outlined on that slide where I walked you guys through it. And you can do this, you can do some practice with some template strands, with some non-template strands. Just remember, you're always going to be using the template strand to make your mRNA. Now, let's talk about what happens if we make changes. Okay, what if we change that DNA sequence? Now, any permanent change to an organism's DNA is called a mutation. Okay, some sort of change in that genetic information. And we often see a change in genotype, which can lead to new alleles, right? New phenotypic effects. Now, there's two major classes of mutations. Point mutations, which is just a small number of base changes. So this is like changing 1 or 2 or 10 bases, or chromosome level mutations. Okay, these are changes to entire chromosomes, so much larger scale. And we're going to talk about each. But we are first going to talk about point mutations. So here is showing you an unrepaired mistake that is leading to a point mutation. So we have your original sequence. We have a base pair mismatch, and it wasn't caught, okay? It wasn't caught with our proofreading. It wasn't caught with our mismatch repair. And what happens when this mutated strand, okay, is replicated, we end up with a new strand, this time without an error, right? Because we used that wrong base as our template. And now we don't even have a mismatch. This is a complete change, a mutation to our original DNA strand. Now, we typically classify our mutations not only by point versus chromosome, but how do they impact our organism? And when we talk about impacting our organism, we have to talk about fitness just briefly. Okay, I think we mentioned it way back in the beginning, but fitness is an organism's ability to survive and reproduce. Okay, so the more fit you are, the better you are at surviving and reproducing. So mutations can have pretty much 3 types of consequences. They can either be good, okay, and they can increase your fitness. They can make you better at surviving and reproducing. They can be neutral. They can not affect you whatsoever. Or they can be bad. Okay, they can affect you negatively. Now, a lot of our point mutations are either going to be neutral or bad. Okay, deleterious. Very rarely are we going to actually see a beneficial mutation. So we're going to talk about 4 different types of point mutations: missense, silent, frame shift, and nonsense. And with these, make sure you can identify them and you know, are they good, neutral, or bad? Okay. So with missense, this is going to be a change in an amino acid that does change the protein product. Okay, you're going to have some amino acid change. We also can have a silent mutation where you get a change in a base, but it doesn't change the amino acid. And this boils down to redundancy. Remember, we can have 4 different codons code for the same amino acid. We also have frame shift. These are going to shift our reading frame, which we'll see kind of what that means, but it's usually either an insertion or a deletion of a base in a series that's not a series of 3. We also have nonsense mutations, which are going to change a specific amino acid into a stop codon. So let's see what that looks like. So our first mutation up here, or our first sequence up here, is our normal DNA. This is what it should look like. Okay, original sequence. We have our original mRNA and our original polypeptide. So this is what we should be making. This is what it should look like with no changes. Now, our next type is a silent. So you'll see, right, instead of a T at this position in our first codon, we have a mutation. It's changed to a C. But you'll notice, okay, our protein, our amino acid, is still tryptophan. Okay, no change. This is a silent mutation. Now, what do we think? Good, bad, or neutral? These are going to be neutral, okay, because we didn't see a change, right? It did not change our protein, and so we're not going to see an effect on phenotype. Nothing's going to change. We have the same protein at the end of the day. Now, next type, missense. So we'll see, here's a mutation. We have a T instead of a G, and it did change our amino acid. Instead of tryptophan, we have cysteine. Okay, so some change to our protein sequence. Now, this can be good, it can be neutral, and it can be bad. It just depends on what that change was. Okay, so this one kind of can be whatever. Next is our nonsense. You'll notice, okay, TGA, we got a mutation here. Should be a G, but it's an A. And what happened is we got a premature stop codon. So now instead of getting a nice long amino acid sequence, we just got one amino acid and that's it. Okay, we just got some tryptophan. Now, these are almost always bad. Okay, they are always going to cut off some part of our protein, and that usually changes its function. Most of the time, it'll shut it down completely. Okay, so this one's pretty bad. Now, last and possibly worst of all, well, most definitely worst of all, is our frame shift mutation. So what you'll see is we actually inserted a single base. Okay, so we had a TGG, and now we inserted a C between that first T and G. And look what it did. Okay, look at our sequence. Is it at all the same to our original protein? No. Only that first amino acid is the same, and everything else, everything downstream, that's what we call it, downstream of that insertion is changed because we changed the reading frame. Right? We changed the codons. We changed our sets of 3. So if we shift that reading frame, all of our subsequent codons are going to be changed. Now, I'll tell you right now, this one's pretty much always bad. Okay, this is the worst of the worst. Okay, you can think of it as kind of increasing severity. We have our neutral. We have, this one could be a few things, good, bad, ugly. Almost always bad. Very much almost always bad. Frame shift tends to be the worst. You very rarely get a functioning protein when you change that reading frame. And even if you get a functioning protein, it very, very rarely can do its original job. So what about chromosome level mutations? So these are much bigger. Okay, these are much bigger in scale. It's not a few bases being changed. Now we're completely changing chromosomes. Now, this can be something like polyploidy or aneuploidy, right? An extra chromosome or one too few. That's an example of a chromosome mutation. Not only can we change the number, but we can change the structure of our chromosomes. So we're going to see things like inversions. This is where part of a chromosome will break off, basically do a flip, and then rejoin. We've inverted a section. We also have translocations, where you break off a section and it attaches to a different chromosome. We can have deletions, where certain regions of our chromosomes can be lost, and duplications, where we can make extra copies of certain regions of our chromosome. So here's what each of those look like. We see a deletion. We've completely got rid of a section of our chromosome. An inversion, we've flipped around. Okay, we've essentially popped off a section, we've flipped it around and rejoined it. This is a duplication. We have an extra section that we shouldn't have in our chromosome. Translocation, so we've had two chromosomes kind of swapping out parts. And these are all examples of these structural mutations. We've changed something about the structure. Make sure you can visually identify each of these. Now, like point mutations, chromosome mutations can be good, they can be neutral, or they can be bad. Now, cancer cells almost always have bad chromosome mutations. Okay, cancer cells can have pretty messed up looking chromosomes. You can see aneuploidy, inversions, translations, deletions, duplications, all of that if you were to visualize a cancer cell's chromosomes on a karyotype. So, for example, take a look at these. Can you tell me what is wrong with the chromosomes on this karyotype? Well, I'd probably start with what is right. Okay, almost everything is wrong. Really, chromosome 8's pair is doing okay. Okay, we want the colors to match. That means each color is a gene region. So 8's doing pretty okay. 20's doing good. Okay, but look at 1, right? Colors don't match. That means that we have either a translocation, something's going on where we got an incorrect sequence where it shouldn't be. Chromosome 2 has 3 chromosomes. This is supposed to be a diploid. We're supposed to have 2. Okay, same with pair 3. We got an extra chromosome. Pair 19, look at that. Okay, we have an extra 4 chromosomes to pair 19. So this is a great example of a karyotype of a cancer cell. You see a whole heck of a lot of chromosome level mutations in these cancer cells.